All right, guys, today we're going to look at organelles. So let me fire this PowerPoint up and we'll get to work. So just a second. All right, so here we go. So here's the deal with organelles. A couple things that we want to focus on moving forward. We're going to look a lot at the structure and the function that goes into the makeup of a cell here, okay? So let me see if I can get rid of that. Nope, can't. All right, so a couple little review things. First, make sure from our other slides we know a couple of these guys. All right, Robert Hooke first got to come up with the word cells. Okay, Robert Brown discovered the nucleus in the 1830s. Schleiden and Schwann and Virchow, right, talked about cell theory. Schleiden said all plants were made of cells. Theodore Schwann says all animals are made of cells, and Virchow says all cells come from other cells, right? So make sure that we know cell theory. All right, that's what is the, I guess, conclusion from those three postulates, those three statements that say all matter is composed of one or more cells. The cell is the smallest unit of structure and function in living things. It's why we don't classify a virus as being alive, because it's not made of cells. And all cells come from pre-existing cells. So that's cell theory. A little bit more review, right? There are two kinds of cells. There are prokaryotes, right? They do not have a nucleus. They don't have membrane-bound structures, organelles. And there are eukaryotes, okay? They have a nucleus. Remember that pro means before and karyote means nucleus, so before the nucleus. And the prefix EU is a Greek prefix. It means true. So a eukaryote, eukaryote, is a true nuclei, okay? Now, both of these have a membrane, right, made of phospholipids. Both have cytoplasm or cytosol, same thing, and they all have ribosomes. However, in a prokaryote, the ribosomes are much smaller. When we look at these kinds of cells through the microscope, we should be able to spot them. The prokaryote on the left doesn't have a nucleus. It just looks like a cell with some stuff in it. There's nothing really definable. But a eukaryote on the right definitely has a nucleus, and we can see that right there. Oh, let's see here. Why is my pen not working? There we go. Okay. So there's our nucleus. Now, I don't want you to think that prokaryotes are not complicated. They're not complex, because they certainly are. All right. They do have parts, right? They do have things that do a job. They have structures that have a function. And, you know, in first year biology, we're not going to look too closely at the prokaryotes, but just as a general overview, they've got a cell wall. Bacteria have a cell wall, just like a plant. Okay. They have a plasma membrane or a cell membrane inside the cell wall. They've got ribosomes, which are going to be used to make proteins. They have what's called the nucleoid, which is kind of like where a high concentration of their chromosomes are. And the bacterial chromosome is obviously in the nucleoid. They can have things called fimbriae, which are like little hairs. They can have flagellum, okay, which help get around. And we're going to look at some prokaryotes, but we're really going to focus on eukaryotes, okay? So let's compare the two. Here is a picture of a eukaryotic cell and a prokaryotic cell. This guy here is a white blood cell, a WBC, okay? That guy, that big fella, he's a eukaryote. He is the active immune response in your body, right? So this white blood cell is looking for invaders, and when it finds them, it touches them with these little cell projections here. And if they don't have the right antibodies, they don't have the right antigens on their surface, then the eukaryotic cell, the white blood cell here, eats them. And that's how it keeps your body safe from disease. Now, these little guys here, this is a bacteria, and that is a bacteria. These guys are in sets of two, it looks like. And they're forming little chains. So these fellas on the white blood cell are going to be absorbed. They're in bad shape. This guy, he still has a chance, but he better run away. All right, he needs to leave. So looking at this picture, it's pretty obvious that cells are pretty small, right? We don't see this with the naked eye when we look at, say, the surface of your skin or into a cut, something like that. And there's an important question there, like why are cells so small? And the answer is pretty simple. The cell volume really matters. So you want your surface area of your cell and the ratio to cell volume to be really small. And here's what that means. The nucleus is in the middle of the cell, right? Generally near the middle. It's controlling everything. If there's too much cytoplasm around the nucleus, it can't control the activities of the cell. You also have metabolism going on, right? Things inside the cell, chemical reactions inside the cell are breaking down foodstuffs, right, in order to make energy. But if they have to travel really far across the cell, well, then you kind of lose some of the concentration of reactants and you start to have some trouble getting your reactions to move forward. And the last thing, and this is probably the most important one, is the speed of diffusion. So we know that things like water or oxygen or carbon dioxide are going to diffuse into the cell. But if the cell is too big, it won't get across the cell fast enough and the cell will die. So basically, here's the ultimate answer. If the cell gets too big, not enough material, things like oxygen, sugar, waste products, can cross the cell membrane fast enough to accommodate the increase in the size of the cell. It would just starve to death or die from lack of oxygen or die from lack of water. So the cell needs to be small. Well, how does that translate into reality? Well, look at these pictures of these two cubes. So the total surface area for cube A here, this little 1 by 1 by 1 cube, is 6, right? We're saying right there that each side is 1 unit long. We can just say centimeters. So this is 1 cubic centimeter, so 1 cm cubed. Now, if I look at B is 5 centimeters tall, and it's a cube, so it's 5 on all sides. So it's 150 cubic centimeters. Here's the exact same box over here on the right, except now instead of being one big box, we have stacked 1, 2, 3, 4, 5 of these little 1 centimeter cubes on top of each other. Now, we're taking up the same volume, but our surface area is much greater. All right, surface area of C is 750. All right, as far as our centimeters cubed, right? B is only 150. So if I look at the surface to volume ratio and compare the two, so that would be the surface area divided by the volume. Well, this is a pretty low surface to volume ratio, 1.2. It's not great. But over here we have 6. And notice something. It's the exact same as the 1 centimeter cube. So even though I've got 750 cubic centimeters of surface area in C, compared to only 6 cubic centimeters in A, I've got the same surface to volume ratio. So what we're showing here is that if you have a bunch of really little cells and they occupy whatever space, the smaller the cells, the greater the surface to volume ratio, the easier it is for things to move in and out of the cell. So we're talking about the cell. Let's look at the cell. The cell has 4 basic parts we should be able to identify just visually. The edge of the cell is the membrane. In the middle of the cell, somewhere roughly in the middle, is going to be the nucleus. It's going to be like a dark sphere, usually. There's the cytoplasm, which is the jelly that connects everything from the membrane all the way to the nucleus. And in the cytoplasm, you have the organelles. Now, I've made this picture as big as the slide. I strongly recommend you print off this picture and the next slide and have them in your notes. Because this is a zoomed in view, right? A labeled view of an animal cell. All right, now that's really important. This is an animal cell. All right. I know it's an animal cell for a couple reasons. Number one, there's no chloroplasts in it. That pretty much gives away that it's not doing photosynthesis. Number two, it doesn't have a cell wall. Those are the 2 big ones, the 2 easy ones. Another one is it doesn't have a central vacuole. Okay. And we'll talk more about the central vacuole on the next slide. But I want you to learn the structure, so the name of the organelle, and its function. So that's the goal of this lesson. So we're going to talk about each of these and see what they do. If I were you, I'd print this out and I'd write on it as we go. Just write the, ribosomes. This is, they're going to build our proteins, right? We'll just write on there what each thing does. That way you have it all in one place. So that's an animal cell. Here's a plant cell. Plant cells don't have lysosomes, which we'll talk about shortly. They don't have centrioles. They don't have flagella generally. And things that they do have that really sets them apart from an animal cell, pretty obvious. They have a cell wall, which is a much more rigid shape that the cell has. And they also have this tonoplast, this central vacuole, okay? Now, this is the plant cell. I would also recommend that you print this thing out and that you write on it as we go. All right. So let's look at these organelles. So let's look at these membrane. Let's look at these organelles. Sorry, and let's start with the membrane. All right. So the membrane is going to separate the cell from its environment. Okay. It's going to be a boundary. It's going to let certain things come in. It's going to let certain things go out. It's going to keep certain things in and keep certain things out of the cell. We call that selective permeability. Now, this is an organelle we've already talked about. Okay. There is a PowerPoint and a lecture in Google Classroom on that. So if you need a little refresher, go check that out, okay? One big thing to remember about the cell membrane is that it's a bilayer, right? And it's got those phospholipids. So if you see these bad boys around, chances are you're talking about a cell membrane. Now, the second thing that is an organelle, so to speak, right, that we want to talk about is the cytoplasm. And it is a substance, like a jelly-like, fluid-like substance, okay, that fills the cell up. It gives the cell some of its volume. It's mostly water, okay? It's an aqueous environment, so it means it's comprised of water. And it's the fluid part of the cell. And there's actually two kinds of cytoplasm. There is the cytosol, which is like the fluidy part. You know, if you see an amoeba moving and you see the cytoplasm flowing, that's the cytosol. And then there's also like a gel version of it. It's a little bit thicker, kind of holds things in place. So that's the outside of the cell, the membrane, and then what's inside the cell, the cytoplasm. Everything kind of floats in the cytoplasm and, you know, moves from place to place in the cell, moving through the cytoplasm. Now, the organelles itself, all right, are pretty important. Number one, the term means small organ. Okay, it's formed. It's a little body, a little compartment, and it's sealed off from the rest of the cytoplasm. It's sealed by its own membrane. It's the exact same kind of membrane. It's important as far as organizing the structure of the cell. So, you know, when we're trying to figure out where things are, you know, we look for these organelles. It kind of helps us figure out where things are happening. They are a way for the cell to compartmentalize its reactions, okay? You know, if you're breaking down food with an enzyme, you probably don't want that to accidentally break down your nucleus. So it's a really good idea. It's a really advantageous adaptation that we have these organelles. And like I said, it keeps the various enzymes separated. Now, here's the big deal. We want to identify the organelles. We want to be able to describe them. We want to be able to recognize a drawing of them. We might want to be able to draw them ourselves. We want to be able to explain their function. So let's look at the first one, the nucleus. It stands out. It's really conspicuous. It's usually spherical, all right, but it can't have lobes or be kind of irregular in shape. It's got three parts. There's the nuclear membrane, there are nuclear pores, and there is chromatin inside the nucleus. If we look at it a little closer, this is the nucleus, okay? On the right, you can see actual microscopic images of the nucleus. The one at the top is the surface, and then using a transmission electron microscope, you can see the pores that are in that nucleus in that second picture. And down there at the bottom, you can see what's called the nuclear lamina, which is the actual membrane itself there. Now, notice that this is in micrometers. All right, that's a really small unit right there. And so these are teeny tiny little pores. But on the left, you can see there is an illustrated version of the nucleus. The nucleus has a couple parts. Right? It has pores here on the outside. You can see them right there. There is chromatin. All right, chromatin comes from the Greek. It means colored threads. Right, and that's actually the DNA. And then in the very middle is this really dark region. It's called the nucleolus. This is a place where ribosomes are made, ribosomes are stored. The nuclear membrane is a double membrane. It's separated by about 20 to 40 nanometers, okay? And that inner membrane is supported by proteins, and that we call it a matrix, the organization of proteins. That's going to give shape to the nucleus, keep it from collapsing on itself. The pores are holes in both the inner and the outer nuclear membrane, about 100 nanometers across. They are comprised of proteins that give them their shape. And this is what allows material to go in and out of the nucleus. And this is something we'll talk about later when we learn about mRNA, messenger RNA. Now, the nucleolus in the middle is a dark, kind of stained area in the nucleus. There can be none in the nucleus. You could have one or two. You could have up to 4 in the nucleus. And this is a place where we store the ribosomes that we use later. Now, the chromatin there, like I said, it comes from the term colored threads. And it's the DNA and the proteins in a loose format. When the cell goes to divide, it will actually bind up and form the cell's chromosomes. But here's something interesting. Right? When we think about a chromosome, we probably think about these little X-shaped chromosomes, right? Something like that. Those are chromosomes. You can't actually read a chromosome when it's all bound up in that X. The DNA is too tight. So in order to read the DNA, to read those instructions, the DNA has to be loose. And so when you see the chromatin in the nucleus, that's actually the DNA that's being read by the cell. Now, as far as the function of the nucleus is concerned, it's the control center for the cell. It houses the genetic information, okay? And it does a couple other things, but for our purposes, that's pretty much all we need to know. Next organelle, the ribosomes. Ribosomes are actually made of 2 subunits, a little bit of rRNA. That little r stands for ribosomal RNA, and some protein. And its job is protein synthesis. Now, when we look at a ribosome, we find them in a bunch of different places. In the cell, we might find them stuck to the rough endoplasmic reticulum, the rough ER. And you can see here in this TEM, this transmission electron microscope image, all those little dots are bound ribosomes. Now, this line that I'm trying to highlight here, right, that line right there, this line right here, right, these are the folds that you see over here in the membrane. And so there are ribosomes that are stuck to these folds. They're called bound ribosomes. They're bound to the membrane. And then in the middle, you have these free ribosomes that are kind of floating around. It does have 2 parts. It has a large subunit and it has a small subunit. And this cleft right here, this little hole, is actually really important to its function. Now, you want to know where you find them. You'll find free ribosomes floating around in the cytoplasm. They can make proteins out in the cytosol, and they have membrane-bound ribosomes. They usually make the proteins that get exported out of the cell. And now that we've kind of talked about these ribosomes, I just want to go back. I want to point out something. So this over here in blue, right, this rough endoplasmic reticulum that is being highlighted there, that's part of this membrane system that is inside the cell. And so the endomembrane system is the system inside the cell that moves things around the cell in something that we call a vesicle, all right? And you can see kind of laid out here how you can transport something from the nucleus through the rough ER, maybe to the smooth ER, to the Golgi apparatus, all the way out to the plasma membrane. So the endomembrane system is connected, and it's kind of like a highway. It's like an assembly line. It gets things from point A to point B. Now, the rough endoplasmic reticulum and the smooth endoplasmic reticulum are 2 of the organelles we want to know. So the ER, as it's usually called, is about half the total membrane in the cell. It usually touches the nuclear membrane, which is important. That means that things can go directly from the nucleus into the rough ER. Again, down here on this, this would be, I believe, a TEM, transmission electron microscope image. You can see all the little membranes right there. You can see that there are there are bound ribosomes on those membranes. And that's why it's called the rough ER. As far as its structure, okay, it's just a folded sheet. It's got all these tubes and they're wrapped around each other. It's very fluid. It can change shape. It can change size. And there are two basic types. There's the smooth ER, which has no ribosomes on it. And there's the rough ER. It has ribosomes stuck to it. So this is where we find our bound ribosomes. All right. Our bound ribosomes are on the rough ER. Now, the smooth ER has a pretty cool job. It's used to make lipids, so it makes fats. It creates more cell membrane. It can be used to store carbs, and it can be used to get rid of poisons. So this is a pretty cool example of a time where we can directly connect the cellular function with maybe an organ in our body. So hopefully in health, you learn that the liver is responsible for detoxifying the blood. That's one of the jobs that it has. If you were to look at a liver cell underneath a microscope, you would expect to see a lot of smooth ER in that cell. That's one of the ways you can identify a liver cell. Since the liver cell's job is to detoxify poisons and the liver's job is to detoxify the blood, it makes sense that the function of the cell is going to be related to its structure, right? Those two things are in a relationship. If you were to look at a heart cell under a microscope, you wouldn't expect to see a lot of smooth ER because, hey, newsflash, the heart cells are not important for detoxifying the blood. It's their job to pump the blood. So in a heart cell, you're going to see a lot of muscle fiber. In the smooth ER, in a liver cell, you're going to see smooth ER. So even though that we're looking at these organelles in a single cell kind of environment, that's how we're approaching this, they actually do matter as far as the overall function of an organ system works. Now, a rough ER has ribosomes. It makes what we call secretory proteins, or proteins that we're going to secrete. And so if we're thinking again along the lines of the liver cell and the smooth ER, maybe our pancreas could have a lot of rough ER because it is responsible for secreting some of our chemical messengers. Sorry, lost my train of thought there. On to the next organelle, the Golgi body or the Golgi apparatus. Its structure is a parallel array of flattened cisternae. All right, that sounds fancy. It looks like a stack of pita bread. Okay, so like think about pita bread. It's got the pocket in the middle of it. That's what it looks like, or like a bunch of pancakes on top of each other. You can have as few as 3, maybe as many as 20 in a cell. Not a whole lot is known about the Golgi. I mean, more is learned, over time, but they do think that it is likely an outgrowth of the endoplasmic reticulum system, so they're connected. So this is what it looks like. Oh, excuse me. Ooh, you can definitely see here this the pita bread thing we're talking about, right? Looks like a pita sandwich. And here it is on our microscope image. And it's got a cis face and a trans face. The cis face, CIS, receives packages from usually the rough ER, maybe the smooth ER. The trans face, the other face, it is going to ship things out of the Golgi. So in the Golgi, things are modified, okay? They're distributed, they're gotten ready for distribution throughout the body. So up here we would have the cis face, and here we would have the trans face, okay? And you can see the trans face looks like this, and the cis face looks like that. Now, as far as its function, it needs to modify the products that are coming out of the ER. So that, remember, the ER is making proteins. It needs to process those, and then it needs to distribute them. It needs to send them on to wherever they need to go. In order to do this, it has little taxis. So these little Golgi vesicles, they're little membranous sacs that bud off the Golgi body, and they basically act like a taxi. They transport things from the ER around the cell. Now, some of those are called lysosomes. A lysosome has a single membrane, and it's made from the Golgi apparatus. Now, the lysosome's job is to house some enzymes. There's a big key here. The prefix lys, L-Y-S, means to cut or to break down. And so if you're going to lyse something, you're going to pop it. You're going to cut it open or cut it down. So a lysosome is carrying the enzymes that are going to cut things. They can be enzymes that cut fats. They can cut proteins. They can cut sugars. They can cut nucleic acids. like 40 different kinds of lysosomes that we're aware of. So let's look at this as a whole, right? We have the rough ER. It's produced something, right? Whatever it is right there in that transport vesicle. It goes to the cis face of the Golgi, where it is then packaged. It leaves the trans face of the Golgi, and these lysosomes go on to do things. Now, there's two things a lysosome can do, can break down. It can break down food particles. So we have phagocytosis right here. Remember, phago means to eat. All right. So if we're bringing food in, we have phagocytosis there. And so the lysosome, which is made of cell membrane, right? That's the outside, and the food vacuole, which is also made of cell membrane, when they touch, they just fuse together. The enzymes enter the food particle and they digest it. You can also do something called autophagy. Auto, the prefix, means self. So autophagy is when you eat yourself. Now, you might be thinking, well, that would be bad, right, for the cell. We don't want to do that. Well, actually, it's actually a good thing sometimes. So this here looks like a mitochondria. Let's say the mitochondria is damaged or it's old and broken down. We don't just want to toss it. It can, we can recycle a lot of those proteins, a lot of those building blocks, and that saves the cell from having to go out and find new building blocks. So you can break down old organelles. You can break down broken parts of the cell. Now you can see with just a little bit better version of what's going on here, just a little more in depth of what we just looked at on these two images. Okay, you can see here, this is like real life, right? This is our TEM. So we have a mitochondria, a piece of it, it's all broken up. We have a peroxisome. So this has hydrogen peroxide in it, which is H2O2. H2O2 is not something you want to get on your cells. It will kill them. That's why we put hydrogen peroxide in a cut, right? It helps get rid of that bacteria. Now, another job of a lysosome is sometimes to actually kill the cell. If you're missing enzymes, that can, you can end up with some kind of genetic enzyme disease, things like Tay-Sachs or Pompei's disease. If you're interested in that, you can certainly look those up. On to the next one, the vacuole. The vacuole is pretty simple. It's very similar to a lysosome. It's a single membrane. It's usually a little bigger than the Golgi vesicle. And the function is going to depend on the organism. So here are two. If you're a protist, all right, you could have a contractile vacuole, which there was a video of one of those earlier in the year that we looked at. This vacuole constantly fills with water, and then when it gets full, it pumps that water out of the cell. You can also have a food vacuole, okay? A food vacuole is going to store ingested food from phagocytosis until a lysosome gets around to digesting it. So there's two different kinds of vacuoles. This was a screenshot from the video that we watched earlier. You can see up at the top, this is the contractile vacuole. It's full of water. And when it contracts, all the water gets pushed out. It helps the cell maintain homeostasis. Plants also have some interesting vacuoles, like the tonoplast. It is a single vacuole that can be up to 90% of the volume of an adult plant cell, of a mature plant cell. It's responsible for regulating the water that is in the cytoplasm of the plant cell. It allows the plant to store ions there, salts, ammonium salts, fertilizer, things like that we might want the plant to use to grow. It's where you store the pigments that give all the different flowers their colors and things like that. Some cells use them to store toxins, okay? And this central vacuole is responsible for what we call turgor pressure. So if the tonoplast, if the central vacuole is full, then there's a lot of turgor pressure. The plant stands up nice and straight. If there's not a lot of turgor pressure, if the central vacuole is pretty empty, then your plant will start to wilt. All right, so it's a pretty important organelle. Here you can see it. There's this central vacuole is pretty big in this plant cell that we've got here. All right, the mitochondria. Now, hopefully, you have heard the joke before, right? The mitochondria is the powerhouse of the cell. It's true. We're going to learn about a couple extra things, though, not just that. First, it's a double membraned organelle. It has an inner membrane with more surface area than the outer membrane. The inner space is called the matrix, and the space between the two membranes is called the intermembrane space. And so on the left, we have an illustration. On the right, we have an actual electron microscope picture of a mitochondria. And you can see that the inner membrane has all these like wavy bends, okay? It's going back and forth and back and forth and back and forth. And here's the deal. These folds increase the surface area, and the more surface area there is on the inner membrane, the more reactions can go on at once in a mitochondria. That's really, really important. So I'm going to say that again a couple times in the next few slides. The inner membrane is folded into cristae. That's what they're called. Those folds are called cristae. If you have a lot of folding, cell is probably very active. If you don't have a lot of folding, cell is probably not active. There are lots of enzymes, lots of proteins inside those membranes. And this is where we make ATP, all right? ATP is the energy molecule of the cell. So very important that we know where that's made. That's why the mitochondria is called the powerhouse. Now, the way that we make ATP is called cell respiration. This is the release of energy from food, namely glucose, okay? The ATP that we make, the majority of it for our cell, is made in the mitochondria. Now, the mitochondria has a couple interesting other facts about it. Number one, it has its own ribosomes, but they're really, really small. The mitochondria have their own DNA. They can reproduce themselves. And when you take those three things together, there's a hypothesis that says they were likely independent cells at one time. And we'll talk more about that later in the year. But the mitochondria are really unique organelles. Something very similar to the mitochondria is the chloroplast. It also has two outer membranes. It has a really complex internal membrane. And instead of the matrix inside, the fluid is called stroma, and that's what fills up the inside of the chloroplast. This is, on the left, an illustrated image of a chloroplast. On the right, you can see the electron microscope image. I think the left one's a little easier to see, okay? But inside your chloroplast, you have the liquid, the stroma, right there, okay? And then there are these thylakoids. The membranes inside are kind of organized into these flat discs. We call each of those discs a thylakoid. If you have a bunch of thylakoids on top of each other, we call them a granum, and that's where we store our chlorophyll. That's where the pigment is actually stored. It's inside the thylakoids. And that's what we just talked about. Sorry, I should have switched my slides. So the inner membrane is arranged, again, into those thylakoids. If you have a bunch of thylakoids on top of each other, we call it a grana, and the green pigment chlorophyll is stored there. Now, you probably know already, chlorophyll is used in the process of photosynthesis, which is the use of light energy to make food. Some other things about chloroplasts: they have their own ribosomes. They're really small. They have their own DNA. They can reproduce themselves. They often have starch, okay, a source of energy for themselves, and they were likely independent cells at one time. You'll notice that the chloroplast and the mitochondria are very similar structures. They both have ribosomes. They both have their own DNA that's different from the DNA in the cell. They can reproduce themselves. So, all of this leads us to look to see where they came from. And they may have been some kind of cyanobacteria or some other form of primordial independent cell. Now, there are also things in plants called plastids. I'm just going to throw them at you real quick because you're going to see those later on just briefly. They're just a bunch of plant organelles. They have a single membrane and they store things. So they're very similar to vacuoles, but they're just in plants. Things like amyloplasts, which store starch, or leucoplasts. There's also chromoplasts, which store plant pigments like carotene. It's what turns carrots orange. There are some other things in plants that we talk about called orgastic materials. They are just generic term. That's the generic term, for substances produced by or stored in a plant cell in the plastid. Things like tannins or latex or resin. Now, I don't know if you know about the last two, but if you are a good old Rockbridge boy and you hunt a little bit, you probably know that tannins are found in acorns, right? And that for human beings, tannins can be toxic, but for deer, who obviously love acorns, they don't really mind that. All right, the next organelle we want to look at is the cytoskeleton. It's a bunch of rods and filaments that kind of form a network inside the cytoplasm. You can see a really zoomed in view of them here on this microscope image. Okay, and they are found in the cytoplasm. So the cytoplasm is not empty. It's not just jelly with nothing in it. There's something there. It's this microtubule, microtubule and microfilament and intermediate filament cytoskeleton. The function of the cytoskeleton is obviously to give structure and shape to the cell, just like your skeleton gives you structure and shape. It allows the cell to move, and it's actually very important for cell division. It's got 3 parts: microtubules, microfilaments, and intermediate filaments, okay? And you can kind of see a breakdown of them here. Now, you don't need to memorize this by any stretch of the imagination. Not going to ask you to do that. One thing I want you to know about each one. All right, the microtubules is a hollow tube. It's literally a tube. That's what lets us maintain cell shape. All right, it's how we build cilia and flagellum. That's important. For the microfilaments, these are actin filaments. Now, when you hear the word actin, think action. Okay, this is what allows the cell to change shape. It's like muscle contraction. It's actually what contracts your muscles. Okay, so this is like a tension-bearing element. This is something that can move back and forth. It can contract. It can, help the cell move. And then the intermediate filaments, okay, they are important for maintaining the cell shape. Okay, just like actin, they can bear some tension, but they're also important for anchoring things. So microtubules, hollow, they make things like cilia and flagella. Make sure you know that. The microfilaments are made of actin, okay? They change the cell shape, they do muscle contraction, and the intermediate filaments, all right, they anchor the nucleus and other organelles in place. All right, so that's the end of this video on organelles. If you have questions, let me know, and I'll be glad to give you some help.